Today we're diving into the incredible world of our cells. Each cell is a marvel of nature, packed with intricate machinery and bustling with activity. Imagine your cells as bustling cities, with membrane proteins acting as gatekeepers, messengers, and sensors. These proteins are embedded in the cell's outer layer, the membrane, and they play a crucial role in keeping the cell's environment just right. These proteins control what enters and exits the cell, from tiny ions to large molecules. They're like security guards, ensuring only the right substances get in or out and keeping harmful invaders at bay. They're essential for life. When you feel a touch or respond to a hormone, membrane proteins are at work. They help your nerves send signals, your muscles contract, and your organs communicate with each other. Over half of all modern medicines target these proteins. From painkillers to heart medications, many drugs work by interacting with membrane proteins to change how our cells behave. Understanding them is key to developing new treatments and cures. By studying how these proteins function, scientists can design better drugs with fewer side effects, and even find new ways to treat diseases that were once thought untreatable. But studying them isn't easy. They're deeply embedded in the cell's oily membrane, making them tough to extract. Their location makes them both fascinating and frustrating for researchers. Their dual nature, oily parts loving the membrane, other parts loving water, makes them stable in cells but unstable in labs. This unique structure is what allows them to do their jobs, but it also means they fall apart easily when removed from their natural environment. Removing them is like studying a deep-sea fish on land. They lose their shape and function. Just as a fish needs water to survive, membrane proteins need the support of the membrane to keep their structure. To unlock their secrets, we need to gently coax them out without destroying their structure. This requires special techniques and a lot of patience, as even the smallest mistake can ruin the protein. This challenge sets the stage for our scientific adventure. Scientists around the world are working together, combining their knowledge and creativity to solve this puzzle. Let's discover the tools that help us liberate these cellular gatekeepers, and see how breakthroughs in this field could change medicine and our understanding of life itself. The cell membrane is a remarkable structure, made up of a lipid bilayer, two flexible layers of fatty molecules that form the boundary of every living cell. This bilayer acts as a selective barrier, protecting the cell's internal environment while allowing certain substances to pass in and out. Each lipid molecule has a water-loving or hydrophilic head and a water-hating or hydrophobic tail. When placed in water, these molecules naturally arrange themselves so that the heads face outward toward the water, while the tails hide away from it, creating a stable, double-layered sheet. Membrane proteins are embedded within this oily barrier, nestled deep among the lipid tails. Their hydrophobic sections are hidden away, protected from the water, while other parts may poke out into the watery surroundings inside or outside the cell. To study these fascinating proteins and understand their crucial roles in cell signaling, transport, and communication, scientists need to extract them from the membrane and place them into water-based solutions where they can be analyzed. But there's a major problem. Hydrophobic proteins don't like water. When removed from their cozy membrane environment, they tend to clump together, misfold, and lose their natural shape and function, a process known as aggregation. This is the central dilemma for researchers. Removing a membrane protein from its natural home in the lipid bilayer often risks destroying its delicate structure, making it nearly impossible to study in detail. To overcome this, scientists realized they needed a way to mimic the membrane's oily environment, but in a water-based solution. The challenge was to create a safe haven for these proteins outside their native habitat. The solution? Find a special molecule that could wrap around the protein's hydrophobic parts, shielding them from water, while still allowing the whole complex to dissolve in water. This would keep the protein stable and functional for study. This molecule would need to bridge the gap between the oily world of the membrane and the watery world of the laboratory, acting as a molecular translator between two very different environments. For decades, this challenge was a major roadblock, slowing progress in the field of membrane protein research and limiting our understanding of these vital molecules. But then, a clever class of molecules was discovered: detergents. These remarkable compounds changed everything, opening the door to new discoveries. Let's meet them. The answer? Detergents, the same kind you use to wash dishes. Detergents are amphipathic. One end loves water, the other loves oil. When added to cell membranes, their hydrophobic tails wedge into the membrane, breaking it apart. Detergents surround membrane proteins, forming micelles, tiny bubbles that shield hydrophobic regions. This keeps proteins stable and soluble in water, preserving their structure and function. Detergents are essential for extracting and studying membrane proteins. They allow us to purify proteins for further analysis like chromatography. Without detergents, membrane protein biology would be stuck. But not all detergents are equal. Choosing the right one is crucial. The art and science of detergent selection begins here. Let's explore the main types. Meet the ionic detergents, the sledgehammers of the lab. Their charged head groups make them powerful at breaking apart membranes. Sodium dodecyl sulfate, SDS, is the most famous, used to unfold proteins for analysis. Ionic detergents are great for solubilizing stubborn proteins, but they often denature them. If you need a protein's function intact, these are too harsh. They're perfect for techniques like SDS page, where unfolding is the goal. But for functional studies, they're a last resort. Think of them as demolition tools, effective but not delicate. Understanding when to use ionic detergents is key. For most purification, gentler options are preferred. Let's meet those next. Non-ionic detergents are the gentle giants, fine-tipped paintbrushes for protein work. Their uncharged head groups make them much milder than ionic detergents. Popular examples include Triton X100, dodecyl maltoside, DDM, and octyl glucoside, OG. They solubilize membranes while preserving protein structure and function. This makes them ideal for extracting active proteins for structural and functional studies. The trade-off? Sometimes they're less effective at breaking up tough membranes. Different proteins prefer different detergents, so screening is essential. Non-ionic detergents are the workhorses of modern membrane protein research. They let us capture proteins in their natural, active state. Zwitterionic detergents offer a middle ground, gentle yet effective. Their head groups have both positive and negative charges, balancing power and mildness. CHAPS is a classic example, useful for solubilizing tricky protein complexes. Performance varies by protein, sometimes perfect, sometimes too harsh. Enter designer detergents, custom molecules like amphipols and MNGs, tailored for specific proteins. These innovations stabilize even the most unstable proteins, like GPCRs. Designer agents are opening new frontiers in membrane protein research. The future is custom-made solutions for the toughest challenges. Next, how do you choose the right detergent? With so many detergent options lining the shelves, it can feel overwhelming to pick the right one for your experiment. Each detergent has its own strengths and weaknesses, and the best choice depends on your specific needs and goals. Start by defining your experimental goal. Are you aiming to keep your protein functional for downstream studies, or do you simply need to detect its presence? The answer will guide your detergent selection and help you avoid unnecessary troubleshooting later. For functional studies, it's crucial to use gentle detergents, such as non-ionic or zwitterionic types. These detergents are less likely to denature proteins, helping to preserve their structure and activity for further analysis. Next, think about the protein itself. Some proteins are robust and can tolerate harsher detergents, while others are extremely sensitive and require extra care. Understanding your protein's characteristics is key to making the right choice. Don't forget to check the scientific literature. Look for published protocols that have worked for proteins similar to yours. This can save you time and provide valuable insights into which detergents are most likely to succeed. Also, consider compatibility with your downstream applications. Some detergents can interfere with protein assays, chromatography, or mass spectrometry, so it's important to choose one that won't disrupt your workflow. If you'll need to remove the detergent later, plan ahead. Detergents with high critical micelle concentration, or CMC, are easier to remove by dialysis or dilution, making them ideal for purification workflows. Most importantly, don't rely on theory alone. Screen several detergents in small-scale tests. This hands-on approach helps you identify which detergent works best for your specific protein and application. Try a variety of types, such as DDM, OG, and CHAPS. Each has unique properties, and you may be surprised by which one gives the best results for your protein. Carefully analyze your results, look for efficient protein extraction, and preserved activity. The right detergent will help you achieve both, setting the stage for successful experiments. Remember, there's no substitute for empirical testing. Every protein is different, and what works for one may not work for another. Careful screening and documentation of your results is the surest path to success in detergent selection. Now, let's see how these choices play out in a real-world workflow, and watch as we put our detergent champions to the test. Let's look at real-world examples. First, the Nobel-winning structure of a GPCR, a notoriously unstable membrane protein. Scientists used DDM to extract the receptor, then switched to a milder detergent, FC12, for stability during crystallization. This combination unlocked the protein's structure and revolutionized the field. Next, the bacterial lactose transporter, LACY. Researchers needed it functional for transport studies. DDM was gentle enough to extract LACY without denaturing it, and could be removed easily for reconstitution into artificial membranes. The right detergent made these breakthroughs possible. Each protein and experiment demands a tailored approach. Detergent choice can make or break your results. Mastering this step is essential for success. Let's wrap up with some final wisdom. Common pitfalls: using the wrong detergent concentration. Too little leaves protein stuck, too much can denature it. Always titrate to find the sweet spot. Don't forget about detergent removal. Plan ahead with high CMC detergents if needed. Watch for interference. Some detergents affect assays or measurements. Always run controls to check for background effects. Careful planning and methodical screening are your best tools. Start with your goal, learn from others, and test your assumptions. The world of membrane proteins is challenging but rewarding. The right detergent unlocks their secrets and advances science. Choose your molecular crowbar wisely. Now go forth and explore the hidden world of membrane proteins.